Good morning, everyone. It's my pleasure to introduce today's keynote speaker, Dr. Zhiping Weng. She's a professor of biochemistry and molecular pharmacology at the University of Amherst Medical School. She is also the director of the program in bioinformatics and integrative biology there. She graduated from the University of Science and Technology of China in 1992 as an electrical engineer. And then in 1993, she moved to Boston University to their program in biomedical engineering, where she completed her PhD in 1997 on calculating binding free energies of protein-protein interactions. She then joined Boston University as an instructor, where she was responsible for development of bioinformatics educational program. And then she was an assistant and associate professor at the same university, and her research focused there on computational methods for the understanding of transcriptional regulation and protein-protein interactions. In 2008, she moved to the University of Massachusetts Medical School to become the director of their new program in bioinformatics and systems biology, and she became a full professor. She has a very broad research interest, and I noticed from her website, in her own words, she says that she's trying very hard not to be interested in everything. And her lab focuses on three main areas, so gene regulation, protein docking, and small silencing RNA. She's also one of the key driving forces behind the Encyclopedia of DNA Elements project, so the famous ENCODE project, where she heads the ENCODE Data Analysis Center, and she coordinates a multicentric team of high-profile bioinformaticians, including Mark Gernstein, Manolos Kellis, Mark Daly, Roderico Gigo, and several others. So the big bioinformatics power behind the ENCODE project, and that's what we will hear today about our work in that area. Zhiping, please, you have the floor.
Works. Thank you. Good morning. Thank you, Eve, for giving the really good introduction. So I'm happy that you all come back after 2 days of extensive meeting to hear about ENCODE. So today I will tell you a little bit about the ENCODE project and what we have learned so far. So ENCODE, as you probably know from yesterday and the day before's presentations, I see a lot of people are using ENCODE data. So ENCODE is a pretty large consortium, roughly with 500 members now. And here is a consortium photo taken from the previous consortium meeting at Salk Institute. So we have a number of goals. And one of the overarching goals is to catalog all functional elements in the genome. And we also, it's a community project, so we really want to develop freely available resources for the research community. That's a very big goal of the whole project. And we focus on human, and we also study other species. Roughly 10% of the ENCODE data are on mouse, and we also have model organism data previously. And the focus of the project is to generate data, to perform analysis, and then deposit the data for community consumption. So ENCODE has been around for a while. Here is a history of the years that ENCODE has gone through. It was initiated in 2003 with a pilot project to focus on 1% of the genome. So many of the students probably here haven't probably started their research career so early on. So at that time, probing the entire genome was really a big deal, and microarray was just available. So it was very carefully chosen 44 regions of the human genome, 1%. And then phase 2 started to gear up towards data production over the entire human genome. And then we added mod ENCODE that had fly and worm, and then later on mouse ENCODE. Phase 3 is the project that I will focus today, and it started in roughly 5 years ago and ended earlier this year in February. Phase 2 started February 1. So every phase, we have had technology development. Obviously, we have data production, and we have computational analyses. And people have been asking me about what's new about ENCODE 4. One thing that's new about ENCODE 4 is previous ENCODEs have heavily focused on data production. Now we have a series of functional characterization centers in addition to data production centers that will try to probe the functions of some of the elements that ENCODE maps. So as I said earlier, we're very community-oriented, so we want to rapidly release the data pre-publication, and we want to provide open access to the software tools and analysis pipelines produced in ENCODE. And to ensure high data quality, we have developed the data standards, QC metrics, and the uniform analysis tools, and all these things are open for public consumption, no strings attached. So one thing that's a new focus for ENCODE 3, which just ended, is we want to provide all the analysis results, all the salient ones, in something called the ENCODE Encyclopedia, which I was charged to develop. And today I will tell you about the encyclopedia. So you probably know this, we really want people to use our ENCODE data, so we keep track of how many publications use ENCODE data and separate them into publications by ENCODE members and outside ENCODE members. And as you can see, even though we continue to publish, the community has found the data useful and start to pick up the publication numbers. And among the publications by outside ENCODE, you can see that people are very interested in human diseases, basic biology, and then also a lot of papers are using ENCODE data for developing bioinformatics methods and tools. So all the data and everything is available from the ENCODE portal, encodeproject.org. So just a couple of slides about my personal experience about being part of a consortium from its inception. So obviously there are lots of advantages. Among them, one is you kind of know where the trend is going, and we have access to a lot of publicly available data sets. Even though there's no advantage of being in ENCODE than being outside ENCODE, actually there are more restrictions about being nice to other members and fair share of data. But it is good to be in it so that you know what kind of data people are working on, and you kind of get ready with your analysis methods. And also, being a consortium, you can generate data using a lot of assays performed on the same cells or tissues, so that enables integrative analysis. You have more uniform data and more of them. And also, the data are of pretty high quality, uniform quality. So a lot of the cleaning is not so painful, and the metadata is very rich, so that you can develop tools, automatically process the metadata, and use them, as you will see, for what we have done using the ENCODE tool, the screen tool that I will introduce later. But one thing you probably noticed that being part of the consortium, and there are a lot of conference calls. I just took this from one of the last phase, and you can see that almost there is a call every single day towards the end of the phase 3. And I think the record was one of the Fridays. We tried to pile up the phone calls on Fridays, and I'll put on my headphone, and I will be on the call for sometimes 6 to 7 hours on Fridays. So it does become painful. One thing that's good about all these phone calls is that I think my English has improved from talking so much on the phone. So what's new about ENCODE 4? Okay, so as I mentioned, we have mapping centers. We have a new thing that's characterization centers. Still, all the data gets funneled into the data coordination center that curates the metadata, controls quality, and then also gets funneled into the data analysis center, which I have headed for ENCODE 3 and will continue to head for ENCODE 4. And we perform analysis with input from community computational groups and put all the analysis results in the encyclopedia. One other thing that's new is that's new about ENCODE 4 is we will take community samples to be processed by the centers, and also we will take community data. As you know, there are a lot of published data out there. How do you get that data into the data coordination center and data analysis center, and then to take advantage of the public data to build the encyclopedia? So just quickly, here are the mapping centers. And as you can see, the type of data we will continue to generate include histone marks, Hi-C, ChIA-PET, TF-ChIP-seq, DNA-seq, and RNA-seq, and then RNA dynamics by, say, incorporating small molecules to track transcription. The characterization centers, there are 5 of them. They probe basically functional analyze and validate regions in the genome with various high throughput ways, such as CRISPR. And the Data Coordination Center is headed by Mike Cherry at Stanford, and Mark Gusting and I co-head the Data Analysis Center. We also have continue to have computational groups that develop novel methods that are not as connected to the data production. So for the rest of my talk, I will have 4 parts. The first part is I will give you a quick summary of ENCODE phase 3 data production. ENCODE phase 3 just finished a few months ago, and we're in the process of writing the consortium paper about to submit. So all the ENCODE data is available from the ENCODE portal, encodeproject.org. As you can see, here are the kinds of types of data we have. And this is probably a handful, but if you, I will quickly walk through these data in a minute. So I just want to make you aware of the relationship between the epigenomic roadmap project and ENCODE. The 2 projects are independent. The epigenomic roadmap project has completed, and all the data that project had are also on the ENCODE portal. We're just hosting the data. So the ENCODE portal has this matrix view that gives you a summary of how many data sets we have. So right now, part of the ENCODE project, both ENCODE 2 and 3 have over 8,000 data sets. And here are the type of assays that these data are on. And if you just look at the phase 3, roughly 5,000 data sets, a bit under 4,000 on humans, and over 1,000 on mouse. And one thing that keeps on coming up is people have the impression that ENCODE data are mostly on cell lines. I just want to point out that a lot of the ENCODE data are on primary cells, and as you can see the next slide, on tissues as well. And actually most of the mouse data in ENCODE 3 are on tissues. So we have a combined ENCODE plus GTEx To form a component of ENCODE data, we call it in-text. There are over 1,000 data sets on 4 human individual donors and across many human tissue samples. And these are not on cell lines, as I said earlier. So one very good component about ENCODE 3 data is this mouse component, and it's very focused on mouse embryonic development. And you can see that there is this matrix of tissue, types of tissues in mouse, dissected from mouse, and then these are developmental stages. And you can see this matrix is almost entirely filled. All the boxes with green color means they have been probed by RNA-seq, 8 types of histone marks, and DNA methylation, whole genome bisulfite sequencing. This is Bing Ren's project. Barbara Wald producing RNA-seq, and then Joe Ecker's lab producing DNA methylation. So some other time points have partial data. And as you can see, this entire matrix would be very good for computational tool development because the data are so even and high quality. So one component I want to shout out for ENCODE 3 is this project by Brent Gravely on RNA binding proteins. And there are over 1,000 data sets. The project has 4 components. One is eClip of RNA binding proteins by Jin You's lab. And also Brent Gravely produced a lot of RNA-seq data while shRNA knockdown, RNA binding proteins. So you can see mostly on 2 cell lines, K562 and HepG2. And then also Chris Burgess' lab produced RNA bind and seq. There is also a localization of RNA binding proteins, lots of images. So that is a summary of ENCODE 3 data production. So at the end, once we have all these data, what do? We want to perform analyses, uniform analysis, and then put the analyses into something called the ENCODE Encyclopedia. So we start from the bottom. You have raw data. And then the ground level of annotation will be things you're very familiar with, such as DNA's hypersensitive sites, histone mark peaks, transcription factor binding peaks, RNA binding protein peaks, all these things all produced from uniform processing pipelines. And then we aim to also perform integrative level of analyses. And today I want to, so for the integrative level, you're probably familiar with chromatin states produced by such as ChromeHMM and Segway, and then also a few methods for annotating GWAS variants, genome-wide association variants, such as HeproReg and RegLonDB and FunSeq, and these are widely used tools produced by ENCODE members. So today I will introduce you to a new tool called Registry of Candidate Regulatory Elements. Some people call it CRE, in short, very dearly. And we have also designed a dedicated visualization and search engine called Screen, which I will introduce in a minute. So the part 2 of my talk is focused on this registry of candidate regulatory elements. So before I get too carried away about the content, let me just tell you a lot of the work has been done by a graduate student in my lab whose name is Joe Moore. And as you know, there are lots of conference calls and lots of things are done by working groups. And there is a registry of CRE working group, and we get lots of help from other PIs in ENCODE. I just want to point out Ross, Barbara, Brett, Tom, and John, they have all given very critical and very supportive input to the construction of this registry. So exactly what do we mean by an registry of CREs? So we want to annotate genomic regions that may function as regulatory elements, such as promoters, enhancers, insulators, repressors, etc. And we want to be broad so that we want to cover data from hundreds of cell and tissue types. We don't want to just narrowly focus on a few cell types. And we want to create registries in both human and mouse. So given those stipulations, we had tried many, many different approaches and settled at the end on something very simple. So ENCODE has a very broad coverage of DNA's seq data across many cell types, cell and tissue types. Okay, so we decided that it's DNA's DHS is known to be high coverage of all kinds of regulatory elements. So we decided to build a representative set of DHSs using all the data across all the cell types. As you can see here, there is a DHS here, which is ubiquitous, and these ones are more cell type specific. So we take the data, roughly 70 million DHSs, and we use an algorithm to iteratively select a representative set to represent the so-called regulatory repertoire of the entire genome. We do this separately for human and mouse. So just as a sanity check, once you get the roughly 2 million RDHSs, you can cluster the tissue sample in ENCODE, a few hundred, 200, 300 of tissue samples by their DNA's signal, and you can see they recapitulate the tissue types as highlighted in different colors here. So they clearly, they are quite representative and tissue specific. So once we have a set of RDHSs, then we want to further annotate them with histomark and TF binding data. So for example, here we have also, we have quite a few types of histomarks, and after analysis and investigation, we settled on 2 very representative histomarks. Adding additional ones didn't really help all that much, so we wanted to stay simple. So H3K4ME3 is a mark for promoters, and H3K27AC is a really good mark for enhancers. So here is a very simple-minded annotation. We want to annotate these DNA's hypersensitive sites to be likely as a promoter, likely to be an enhancer, likely to be bound by the insulator binding protein CTCF. So I just want to point out that this is obviously a very simple classification. It's only for entry-level analysis. There are a lot of enhancers known to, actually a lot of, not a lot, but like a few % of the promoters have been shown to function like enhancers. And then a lot of the enhancers we annotate that are proximal are probably novel promoters. So keep in mind, this is just for simplicity. So in order to annotate those classifications, we use these 3 types of additional data. So obviously, you can see each CRE, so those representative DHSs that is further supported by either one of these 3 types of data will be promoted to the class of CRE. So each CRE could take on any of these colors, as you can see here, using signal peaks. And here is a distribution with respect to the nearest transcription star site, TSS, so whether or not they are proximal or distal, and how many of them. And based on this set of annotations, we annotate the CREs for each individual cell types we have. So roughly 650 of them. So as you can see that these groups segregate according to the epigenetic signal I just mentioned, and in addition, we want to be sure, so we also perform validation using additional data such as Pol2 binding, the binding of EP300, which is an enhancer binding protein, and RED21, which is a cohesin component that also co-bind with CTCF. So as you can see, with a similar data type, they validate into the axes of promoter-like and enhancer-like. So at the end, we classify in a simple-minded manner, we classify our CREs into promoter-like, enhancer-like, CTCF only, and DNase only in each specific cell type. And they have characteristic signal distributions. But as I said, the situation could be more complicated. If you want to delve into more details, you could actually look at each individual pattern for each CRE. So in total, we have curated 1.3 million CREs in humans, and we have less diverse data in mouse, so it's only 430,000 CREs. The human CREs roughly occupies a quarter of the human genome. These CREs on average are 350 base pairs long. So as you can imagine, the conserved or functional bits of it is not as big as 350. And for mouse, it's roughly 10%. So we actually validated a portion of the enhancer-like regions using our approach by Len Panakios' lab using mouse transgenic assays. Using a prediction manner, we actually predict the regions, and then his lab performed experiments to show that a pretty large number of them validate. So obviously, when you have a registry, the one question that pops into mind is how comprehensive is the registry? So if you have a new cell type, would the registry cover the regions that might be regulatory in the new cell type? So what we did was, for some of the cell types in ENCODE, we only have histone mark data or CTCF binding, but no DNase data. So as you know, that the registry is anchored on representative DHSs, so these cell types represent new cell types for the registry. And you can see that the registry has a very high coverage for these new cell types, roughly in the high 80s and 90%. So part 3 of my talk, let me move on to this tool called Screen. So once we have the registry, you know, obviously we can put it on the ENCODE portal, and you can all download it, and you can all write your programs and incorporate it into your analysis. But because ENCODE is very service-oriented, we're interested in serving people who don't program. So one thing that in order to make this registry more useful for those people is to have an engine that puts all of our annotations all together in one place. So this may sound pretty easy, just make a database. At the beginning, I thought, oh, okay, so this is the last 10% of the effort. But actually, it turned out to be a huge amount of effort because we are talking about visualizing at the individual CRE level, and we actually are visualizing roughly 1 terabyte of data that's purely on annotation, processed data. 2 students, MD, PhD students in my lab, Michael Pakura and Henry Pratt, they were my hero for ENCODE 3, and they come up with this screen after like 4 or 5 rounds of design to make this thing actually quite useful. So you can see human and mouse, and basically it's a CRE-centric engine that visualizes individual CREs and all the underlying and associated annotations. It's ENCODE data-centric for now, but we do pull in community data already. And one application is to use these CREs to interpret GWAS SNPs. So if you search anything, you get a table of CREs, and here are the characteristic histone mark and CTCF binding data, and then there are genes, and you can choose them. So we're operating roughly with 2 million such elements together with human and mouse. And you can look at each cell type you're interested in out of 650 cell types. And if you pick the cell type, it will react very quickly to show you the data in that specific cell type. And for each CRE, you can look at where are the genes nearby, are the CREs nearby, GWAS SNPs nearby, genes in the same TED, other CREs in the TED. And you can also see which transcription factors bind to the CRE in which cell type, and also which histone marks are observed in the CRE, in which cell type. Everything pre-computed, 1 terabyte of data, all pre-computed, and then whatever you want, you can just put it up real time. This is one example of incorporating community data. So Systrom database from Shirley Liu's lab has processed all the DNAs and ChIP-seq data from GEO, and over tens of thousands of data sets. So we, any CRE that overlaps with any of the peaks from Systrom will be displayed here. As you can see, thousands of data sets. So it's more, it's an expansion of ENCODE data. So obviously, people are interested in genes. So you can pull up with a CRE which genes nearby and the tissue-specific expression of genes. And also, we have rampage data in ENCODE which shows transcription start site specific expression. So you can see if you change the transcription start site, and different transcription start sites sometimes have very different tissue-specific activities. So we have a registry for both human and mouse. Obviously, we're interested in the orthology. Roughly a quarter of human CREs have an orthologous mouse CRE. And you can directly link that by this orthologous panel. And I think a lot of people want to use, want to browse the genome browser, and then it's very, very slow when you have so many cell types, and the page scrolls over. So we decided that we will crop out the region of the CRE, the signal, and then put them, pre-compute everything so that when you need a region, you can just show it in the cell type you are interested in. And in total, there are 1.1 billion such mini peaks for humans we have cropped out and then pre-computed, and then roughly 10 times smaller for mouse. But it's all there, and you just need to pull it out. You don't need to go to the genome browser if that's a quick view you want to have. So I mentioned earlier, ENCODE 3 has this mouse embryonic development data set. So that it's roughly 70 data sets with very complete assays. So you can compare between 2 tissue and 2 time points, the combinations of them. And you can see here, if you pick 2 time points or 2 tissue types, and you can compare differential gene expression, all pre-computed, and also differential CRE activities, as indicated by red and yellow dots. And the blocks are genes, annotation at the bottom. So the last part of my talk is, one big application from the publications. You can see that people are interested in human diseases. So we, just as a tool specific, we have pre-computed everything with Caucasian population, the GOR's catalog, and then loaded all the data into screen so people can just browse them without doing any calculation. We put all the SNPs and the SNPs in LD, linkage disequilibrium, and we suggest cell types that might be most relevant based on K27 AC signal enrichment. So here are 3 examples. For example, QT interval, the most enriched cell type would be the cardiac atrium. This is actually tissue. So for LDL cholesterol level, you can see the liver comes up as the most enriched cell type. And then ulcerative colitis, you have colon and mucosa of rectum as a tissue that come up. So they are all there, and they're ranked, and you can pick and choose. If you pick a tissue, for example, for QT interval, the particular cardiac tissue, and you get all the CREs, and then the overlap SNPs pull up in tables, and you can drill in each one of them. So let me just give you a couple of examples. One example is to find map GWAS variants for a disease, schizophrenia. So there are 2 studies that have reported one particular SNP here that's associated with schizophrenia. Unfortunately, this SNP does not overlap with a CRE. So what do? So we hypothesized that it must be likely that it's another SNP that's in high linkage disequilibrium with this SNP that has driven the association. So what can you do with it? So you can look at these SNPs about the activity across the tissues in ENCODE. Okay, so among the SNPs that are in LD, and only one of them actually has activity in neural cells and bipolar spindle neuron. So if you delve in with the mini-peak feature I showed earlier that we crop out from the genome browser, and you can see that this particular SNP, that's LD SNP, has high signal in eye retina and has high signal in early brain time points. However, we don't have a lot of human brain tissue, so that especially fetal brain. But as I mentioned, there is this developmental stage data set in mouse, which has a lot of brain regions, as you can see, time point, embryonic stage, 11.5, all that from mouse. So we can take advantage of the human mouse authology. So if you look at the mouse data, this particular orthologous mouse CRE is actually very interesting. It's very high in H3K27 signal, H3K27AC signal in one developmental stage in the middle of embryonic development, and it actually tapers off upon birth. So if you further look into it between human and mouse, and you can see that this region, human is here, mouse is here, good orthology, good syntony, and then also conserved across mammalian sequences. And you can see that this SNP is in the middle of SP3 motif, and the change actually is part of these the nucleotides that contribute to the signal of the motif. So this is a short example to show that we can use the CRE, the registry, and CRE to be a hypothesis generating engine to make predictions to say that this CRE may be, this SNP may be active during human brain development, and then it might be disrupting an SP3 binding site. So all these, now this is experimentally proven, but as you can see from 1 billion terabytes, 1 terabytes of data of annotation, you can generate a lot of hypotheses, and we hope that people who don't have programming skills can just get at all the annotations we have computed and start to use the information to guide their experiments. So let me just give you a conclusion of the parts 2 to 4 that I have told you about. We have integrated the ENCODE data to generate the registry of CREs. And we have also a dedicated web-based engine that allows users to explore and visualize CREs and all the underlying annotations and raw data. You can actually directly go to the genome browser from screen to look at the raw data. The registry of CREs and screen can be used to annotate genetic variants associated with human diseases. And I want to say, here's my last point. I want to put it out, which might be controversial. So my experience with all these analyses is we should actually compute first and ask questions later. Because if you have an idea that you have applied to one particular locus in the genome, you should really apply it to the whole genome and pre-compute and put all the analysis results up so that other people can look at it for all other loci and other cell types that you have data for. So I just want to thank people who have done the work. Obviously, I'm very privileged to stand here representing an entire consortium work over 5 years. And I'm very proud of being a member of the consortium, and we want to be useful, and we want to support other research. And people in my lab who are the heroes are these actually 3 students who have done a lot of the work, obviously guided by other ENCODE members and PIs. So that's all I have to say. And thank you very much for your attention. I'll be happy to answer questions.
Hi.
All right, let's take some questions. Yeah,
Can you hear me?
Yes.
Thank you very much for a great talk. So we are heavy users of ENCODE, and I want to give props from ENCODE 3, especially the mouse development data. And I wanted to ask a question about ENCODE 4, or maybe it's my wishful thinking. So as you know, the standards for biological replicates have been 2 for a while, which enables a lot of integrative analysis, but does not enable differential analysis statistically. Is there any plans on at least of providing increased number of biological replicates for at least a subset?
Yeah, that is a very good question. So ENCODE 3 has shifted its focus away from cell lines to tissues, and tissue samples are precious. So it's actually difficult to even obtain 2 biological replicates for some of the tissue samples. So as far as I can tell, we don't have the push to go for additional biological replicates in ENCODE 4. But if you're interested in, say, human tissues and more biological replicates, I'm actually involved in also another project called the Psych-ENCODE project. It has a lot of tissues, sometimes dozens and hundreds of tissues that you might want to look into there for data for more statistical power.
Thank you.
All right, right at the back.
Yes, I see you.
Very simple question. Well, I looked at your screen tool, which looks really, really cool. But I see that all the annotations is based on HG19, which is the case for most of the data in ENCODE. So what's the plan about using HG38?
HG38 is coming. We are in the process of putting it up. Yeah.
All the data?
Yeah, all the data in HG38. So the thing is, it's a consortium, so it's like a huge truck that moves with a huge inertia. So all these data are processed at the data coordination center. The pre-processing, like uniform processing pipelines. So they compute everything with the same way. As soon as we get that, within a month, the new version will be available. So we're in the process of submitting the manuscript and hoping as soon as we submit it, we will put up the 38, waiting for review.
Left at the front.
As you design your system, how do you balance complexity, functionality in one hand, and ease of use in the other hand, given that you have a broad range of potential users?
That is an excellent question. So I really, learned a tremendous amount from designing a tool like Screen. Obviously, it's an academic product. It's nothing comparable with, say, a commercial product. You're looking at graduate students' work. So we were very naive at the beginning, but during the whole process, ENCODE, the consortium, is really good as part of the consortium because people are very critical. And we have so many testing sessions, alpha testing sessions, beta testing sessions, community testing sessions. We recruited all kinds of collaborators to come and use it and tell us this thing actually sucks, and so you can improve. So we have a lot of rotten eggs on our heads for a while, and we had to let go a lot of the functionality for utility, and it's a balance, and if things are too complicated, nobody understands it, nobody's going to use it. So, it's just a lot of feedback and changes. And if you have anything that we're not doing right and you think we can improve, please let me know.
All right, left at the back.
Hi, are you working on an answer to target gene assignments? Because often in the screen tool I've seen the nearest gene is reported for an answers, but very often I'm interested in knowing what could be the target gene. And since you have the ChIA-PET data, is that something that you are?
Absolutely, yes, yes.
And
so yeah, we have been working on it, so we have an algorithm. So I would say a lot of the ideas have been put out by earlier ENCODE publications. So as you probably remember, this correlation idea of activities across different cell types appeared first in the Manolis Bernstein, Brad Bernstein Nature paper in 2011, even though I wanted to pull up the figure to show yesterday, but the figure is a bit obscure. So the idea has been around in ENCODE Consortium for a long time, and people are, how do we say, you know, it's a prediction. You can base it on correlation of activities. You can base on more sophisticated computational methods. But for the first version of the registry, we want to put out things that are very data-driven, but not so much prediction-driven. And target gene prediction is not yet as reliable. So we will put it up based on high-C ChIA-PET data, and then also based on annotation. We'll tell you exactly what it is based on so that you know what you're getting.
And follow-up question, have you considered promoter capture IC as well?
It's not part of the production effort as far as I can tell. But you know, as you can tell, Bing Ren's lab has made a lot of production, promoter capture, has high-C data already published.
Thank you.
Right at the back.
Hi. Is this on? Yes,
Yes.
okay. I want to push back a little bit on the HD19 versus HD38 question. I'm coming from the perspective of working with a consortium that has 10,000 exomes that have been sequenced and mapped to, you know, over at the Broad Institute to HD19. And, you know, we also work with 100 samples there, 300 samples there, all of which have been mapped very carefully and then curated to HD19. And these are by groups that have had NIH investment to do that, but only to do it once. So if ENCODE does shift to HD38,
It will be back
we have
compatible.
a whole world of data
Yes.
that needs HD19
Correct.
to continue to be maintained.
Yes.
So from my perspective, I see 19 as a more important first mapping for ENCODE and the HD38 as a secondary mapping. And I wonder, you know, that's actually not a great world to be in. We always would like to be in the latest version of the genome. But we're now in a world where we have enormous amounts of computational investment that would be ridiculous to try to redo to lift from HD19 to anything. What do you see as a way to deal with this as a community going forward?
Yeah, we will continue to maintain 19. Whatever we do, it will be backward compatible. And we see exactly the same argument. When we started, everybody was still on 19, even though we know 38 is a much better genome, and it has been out for a while.
18 to 19 was a huge jump.
Yes.
19 to 38, probably incremental, probably not worth redoing yet.
We will redo, because some of the regions, we can tell, I don't know if you use the blacklist.
Yes.
In ENCODE,
Yep.
the blacklist is much, much smaller than 38. That's an indication that 38 has...
The only problem with that is because we have this world of data on 19,
Correct. Yeah,
we
I hear you.
can't do anything with a blacklist in terms of studying various diseases where we have thousands of people whose data is on 19. I actually see that as the bigger problem. I'd love to see everything move to 38 and then to 40 and so on. But we as a community need a way. This is a major high-performance computing challenge.
Yes.
And you know, it's distributed data, it's slightly curated data. We need ways to move back and forth between different genome releases and to take those unmapped reads, many of which aren't even deposited. You know, so the reads that were unmapped to HD19 lost to posterity.
Yeah, I hear you. 19 will be here to stay.
All right, right at the back again.
Hi. This question I want to ask you, I've asked a lot of people, and there are a lot of different answers. But since you've cataloged all the data, I'd like to hear your perspective on what percentage of the human genome is functional.
That is such a charged question, and
Yes, it is.
such a
But you have the data.
question. So as I mentioned, 25% of the human genome is part of the CRE, but the CREs are on average 350 nucleotides long, and you can imagine not every nucleotide is functional in the evolutionary sense, that evolution doesn't care about every base of the 350. So I would say if you imagine is like the middle, like 50 or 100, you can see a portion of the 25% being functional. So I would say 10% would be my guess.
10% of the 25%?
No, 10% of the human genome.
Of the whole genome?
Yes.
Okay. Thank you.
All right, right at the back again.
This is such a great work. The question is, do you think about doing small RNA-seq also next to RNA-seq just to capture microRNA expression?
Yes, I'm sorry I didn't feature that work. So, so there has been a lot of small RNA, microRNA specifically, microRNA-seq data. Ali Morojari has a lot of the data sets in ENCODE for microRNAs, and you can use it, and it's going to continue to encode for.
So it's going to continue. That's
Yes,
great.
yes,
And
microRNAs.
next is, regarding functional analysis, do you thought about doing par clip and hit clip for Argonaut, which actually gives you the target of the microRNA on the gene?
Yeah, that's a very good question. So I have to look across the RNA binding proteins. There are a few hundred of them done by Jin You's lab, whether or not the Argonaut proteins are among the list. It's only E-clip. It is not parse clip and these other more exotic kinds. And that, unfortunately, the RNA binding protein project has not been continued as of yet for ENCODE 4. I know they resubmitted their proposal, so they might be re-included later. But I would say the short answer to your question is unfortunately no. I do know, I do perform research on small RNAs, so I know what you're talking about. And we probably will not have the right kind of data for what you care.
Okay, thank you.
All right, right at the front.
Hi. Hi.
Yes.
My question is, when you have hundreds and thousands of sample sequencing data, when you align to HG19 or HG38, what's the percentage of data that cannot be aligned to that?
That is a very good question. It depends on the data set.
I ask the question because when we are working on, we are creating a genome for a species instead of an individual. So when we do that for a model organism like rice, the genome for a species is much, much bigger than a genome, an individual genome.
Correct.
So that's my ask. Since you have thousands of data, so I ask, what's the percentage of the data that cannot align to the reference individual genome now?
So some of the data, some of the reads that do not align are probably, the reads you don't care about. So are you asking?
Well, you don't know.
We don't know, correct.
Yes.
Could be the pathogens invading your sample. So I don't have an idea. Depends on the data set. For example, like some of the ChIP-seq of histone-mark data sets, I have seen like 90% of the reads map. But for some other data sets, maybe 70% of the reads map. But I think you're trying to ask a question that's more sophisticated than my answer. Okay. So you're saying, what's the difference between individual genomes versus the species genomes? So for the NTAC sample, which might be of interest to you, we are producing individual genomes for those 4 donors, 4 individuals. And you might look into those data to address your questions.
Thank you.
Okay, I have maybe one last, maybe tongue-in-cheek question.
Okay.
So Jorge Luis Borges has this very nice one-paragraph short story about this civilization that is obsessed with the art of cartography. And they make more and more and more precise maps, until at some point they make a map that is as big as the entire country, and people revolt and kick the maps away, and they're done with the art of cartography. So as we produce more and more data, how will we at some point avoid being drawn in all this data and still be able to make sense of that data?
Yeah, I think that's a challenge for all of us sitting here, isn't it? So how do you compromise between simplicity, knowledge, and data? How do you go from data to knowledge and to maybe principles in biology? We have struggled mightily for the Registry of CRE project. At the end, we have roughly 2 million bits of the genome we have settled on, but you know, it's a really, really like, you know, good question. How do we make the compromise? And that is also one of the reasons that ENCODE has included these functional characterization centers, because you can only be a cartographer for so long, and you really want to go and pinpoint each of the landmarks you have picked out, whether or not they're truly guiding the travelers or they're as just biochemical signals.
All right, thanks again, Zhiping.